Misfolded proteins are silently killing your neurons right now. We accept aging as inevitable, but this research challenges that quiet failure mode. Imagine cellular machinery failing to clean up its own junk, causing toxins like amyloid beta to clump into dangerous plaques inside delicate brain tissue. This is the fundamental threat driving neurodegeneration in old age. But there is a sophisticated emergency crew already operating within every cell: heat shock proteins. These molecular chaperones are nature's dedicated garbage collectors. They intercept damaged proteins and keep them folded correctly, preventing aggregation before it starts. The problem we face is not just the plaques themselves. It is our inability to reliably tell these chaperone systems when or how strongly to activate in non-diseased aging cells. Optimizing this proteostatic resilience could redefine what healthy longevity means for your brain health. When cellular maintenance systems start failing, it is often because of an overwhelming load, like cumulative oxidative stress or metabolic strain. Neurons operate in environments that are inherently high energy and prone to reactive oxygen species production. This constant pressure demands peak performance from those chaperone proteins we discussed. The critical question researchers are now asking is not just how HSPs protect against plaques. It is how do we measure the current capacity of a person's proteostasis system when they appear perfectly healthy? We need measurable biological markers that act as early warnings, quantifying the cellular resilience before decline even begins to show up on standard clinical tests. But what if the real failure point is not just toxic aggregation? What if the breakdown begins much deeper, at the core energy factories of the cell? We are finding connections between this protective HSP activity and mitochondrial function itself, the engines powering every neuron. When mitochondria become compromised by oxidative stress, they flood the system with metabolic debris that overwhelms those delicate cellular cleaning crews we talked about earlier. It forces a cascade called unfolded protein response, or UPR, which is nature's emergency shutoff switch for protein folding failure. The real mystery now: how do you measure if your cell's energy output can sustain its own repair crew when under stress? Forget the theory of protein damage for a moment and look at what is happening inside your cells right now. Researchers are finding that we can actually quantify this cellular resilience by looking at specific markers of stress response proteins outside the neuron itself. Think about it. One study showed measurable shifts in blood biomarkers related to chaperone capacity correlating with longevity factors up to 20 years later. This suggests we do not need a biopsy. We might be able to read your brain's defense status just by looking at common lab tests today. But here is the truly staggering part: the potential range for intrinsic neuronal protection, when optimally stimulated through natural means, could increase life expectancy by nearly 25% in certain populations. To maximize this built-in defense system, you need triggers that are precise and systemic, and they require incredibly delicate biological tuning. What exactly activates those mechanisms? The key to achieving that optimal state is not just generalized stress. It is about finely calibrating internal signaling pathways right at the cell level. We are talking about moving past simple antioxidant strategies into complex molecular conversation between energy supply and repair demand. When a neuron senses an overload, it sends specific metabolic distress signals, chemical alerts that initiate targeted chaperone production only where needed. This precision is what we lack in current wellness protocols. Most approaches treat the symptom without speaking the cellular language. We know these stress modulators exist, but they are profoundly difficult to stabilize and apply systemically without unintended side effects on normal metabolism, leading to a hidden risk of metabolic collapse over time. The answer lies in resetting the fundamental energetic feedback loop that governs their entire operational cycle. But this energetic regulation does not happen deep within isolation chambers. The signal to activate these defense mechanisms originates much higher up, linked directly to our metabolic state and endocrine system stability. What we now observe is that HSP activation capacity is not solely a matter of cellular damage. It is actually regulated by the speed at which certain molecules, metabolic intermediates flowing through your cells, can cycle. Imagine this protective crew needing perfect fuel timing. If that internal biochemical flow falters, even if there are no immediate plaques forming, the system operates in neutral, incapable of rapid response when true stress inevitably hits. This metabolic gating function is the key variable everyone overlooked until now. It raises a terrifying clinical question. Can our current markers accurately predict this underlying regulatory failure before physical symptoms appear? The science points to this final, clear verdict on cellular longevity. We are no longer simply talking about fighting plaque buildup. We are learning how to maintain perfect internal flow through that metabolic gating function. The most immediate actionable takeaway is recognizing your body's capacity for rapid repair. Instead of broad supplementation, focus entirely on optimizing the speed and efficiency of energy intermediates circulating in your system. This subtle biological tuning determines whether those protective chaperones can spring into action precisely when stress hits. That mechanism requires precision engineering of your metabolic state itself. If you want to see the full data supporting this entire research archive, including specific figures and citations, visit jmthomasofficial.com. The biological picture is now complete. True longevity resides in managing metabolic timing, not merely mitigating damage afterwards. We discovered that the fundamental capacity of your neuronal defense system hinges on maintaining perfect internal flow through those energetic pathways we studied earlier. The final verdict allows us to shift focus entirely from simply blocking toxins to engineering superior readiness within your cells, ensuring your protective chaperone crew is always optimally fueled and primed for action, whether facing stress or senescence. If you want to examine the full scope of this research archive with all associated figures and citations, visit jmthomasofficial.com.